Well, thank you all. This is so exciting. I didn't expect this many people. And I see Erin, some students I know. So congratulations on putting together our first STEM club. It's very exciting. Yes. So I'm going to talk about telomeres, telomerase, and aging, something that I've worked in the field a bit and recently brought actually to the TMCC campus. So we actually have students now working in this field. So it's pretty exciting. Give you some background and show you some data and a little bit about what we're doing here at TMCC. Okay? So what is aging?
Get old.
Get old? What does get old mean? What is aging?
You have white hair.
You have white hair? Absolutely.
Wrinkles.
Wrinkles? What?
Face falls off.
Yeah, your face falls off. Right? Okay? Yeah, there's a lot of things that we think of as aging. I put together a list of things, right? Not only things we see, but in reality, aging is about some degenerative diseases that don't really afflict us when we're young. And whoa, what happened? One day, it's a problem. Okay? So let me ask another question. What causes aging? Why do we age? Cells get old. That's a great answer. Okay? Isn't this amazing? I'm sure this is a cleaned up photo, but 8 years. Being the president apparently ages you. And you can see that in our president today too. Okay? So why do our cells get old? Why do we age? Lots of different theories, right? And so what I'm going to talk about today is one of the theories of aging, and probably one that if you think of a whole bunch of different reasons why we age, and they're all sticks of dynamite, right? Which one are you most worried about? The one with the shortest fuse, right? That's the one that's going to get you. And science now believes that telomeres are the shortest fuse. So we'll talk a little bit more. But you're right, it's because our cells age. And that's why we get older. If you work with cells, especially in culture, you can easily see the difference. The slide on the top is showing young fibroblast cells, and on the bottom, old fibroblast cells. And they just kind of don't do very well. So what if there was a way that we could prevent these things we've been talking about, or maybe even reverse them? Good thing? Bad thing?
Good thing.
Generally a good thing, right? Especially the older we get, the more we think it's a good thing. Okay? And that's a really intense ethical question, right? It's a philosophical question, and I'm sure we're going to all have time to debate that. But in the short term, then maybe helping with some of these I think would be cool. So bear with me because I'm cumbersome in doing this, but I have a cool video.
And now, eternal youth. Is it in a cage around the corner? News tonight of a breakthrough for some pioneering mice. But we always wonder, what does a fountain of youth for rodents reveal for humans? Here's Sharon Alfonsi reporting.
I feel tremendous.
In the movie Cocoon, it's a swimming pool that turns back the clock for a group of senior citizens. But now, researchers have found a way not just to stop, but reverse the aging process. The key is something called a telomere. We all have them. They're the tips or caps of your chromosome, seen here in yellow. This is what it looks like in a young adult. But as you grow older, the telomeres become damaged and frayed, and as they stop working, we start aging, experiencing things like hearing and memory loss. Scientists took mice who were prematurely aged, added an enzyme, and essentially turned their telomeres back on. You can see it before the enzyme, after.
Their brain function improved, their fertility was restored. It was a remarkable reversal of the aging process.
Look at this picture. The mouse on the right has bad skin, gray hair, and is balding. But the one on the left had its telomeres flipped back on.
And you could see that essentially you now have a dark coat color, that the hair is restored, that the coat has a nice healthy sheen to it.
Even more dramatic, the change in brain size. This is before. The mice had 75% of a normal brain, like a patient with severe Alzheimer's. But after the telomeres were reactivated, the brain returns to normal size. As as for humans, while it is just one factor, scientists now say by looking at our blood cells and measuring those telomeres, you can get a better idea of how well you'll age. The longer the telomere, the better the chances for a more graceful aging. But as for tinkering with them and turning back our aging process, researchers say we still have a long way to go. Sharon Alfonsi, ABC News, New York.
Pretty cool to think about, though, huh? Oh, who knows? Science doesn't like go day by day by day, right? It goes day by day by, whoa, way over here, right?
Better happen before the Daily Judgment, I think.
Yeah. There's many people that are very much about getting this taken care of and spending millions of dollars, because proof of principle experiments have showed that it can work. Right, so let's talk a little bit more first about me. I'll introduce myself really quickly. Okay. My academic journey, I started at a community college right out of high school and did my 3-year and got my AA degree in liberal arts. I was going to become an instructor, a teacher. And I was married. I got married when I was 19. And I lived in California, but there wasn't a university by me. It was the closest one was like an hour away. And so it was real easy, because I was kind of scared anyways, to just say, you know, it's too far, I can't afford it. My husband and I both can't go. And so we both actually stopped school then, and I worked for 10 years or so. And then I was like, well, I really always wanted to be a scientist. I always really wanted to be a teacher. So I said, I better get myself back to school. So I went back to community college, earned my transfer credits, and transferred to the university. And I then got a bachelor's in nutrition science, and then kept on going. Since I wanted to be at a college or a university, that was real important to me. And so I did my doctorate work in environmental science and health, and I focused on the effects of quinone compounds on mitochondrial bioenergetics and mitochondrial DNA. So I had a real diverse, interesting experience there. Then when I graduated, I did a small postdoc, and I thought, well, you know, I didn't learn enough. I want to know more. So I actually got a job at a biotech company here in town, Sierra Sciences. They've been in town since 1999, and they started out at UNR as an incubator program. So I was there for a couple years. After a couple years, I became the director of cell biology. And in a couple more years, I became the vice president of research and discovery. And I had this really great opportunity to learn so many things. But I really always wanted to be at a college, and particularly a community college, because I felt so strongly that what I learned at community college, and I don't mean this in a terribly bad way, but just the university was so much easier for me. Community college really challenged me. I felt like I really learned a lot, and I felt my education was really, really good. I wanted to be back in a community college. So 8 years ago now, I came to TMCC as the lab coordinator, and I've had the opportunity to also serve as instructor in biology and nutrition courses. So enough about me, let's talk about the science. Okay, so the telomere, you guys know a little bit already. It's a nucleoprotein, meaning it's made up of nucleic acids and proteins. Okay, and it's found at the end of our chromosomes. You can see chromosomes up there. The nucleic acid part of it is DNA, obviously, and the sequence is TTA, GGG, and that's repeated over and over again at the end of our chromosomes, which is basically our DNA, right? So at the end of our DNA, and one way to think about it is kind of like caps on your shoelaces. What happens to your shoelaces if you lose your caps? Okay? Yeah, they start to fray, they lose their integrity, so the telomere helps with that. What we do know about telomeres is that every time a cell divides and copies its DNA, okay, then our telomeres get a little bit shorter. In addition to natural process through cellular division, telomeres also can break and fray. And what we know is that when telomeres get too short, a cell quits dividing and starts aging. So the telomeres, in a sense, are like a molecular clock inside of our cells, and the cell is fine to divide until those telomeres get too short. And at that point, the cell signal to no longer divide and starts aging. Okay. So here is a graph showing base pairs, which would be nucleotides, and age here, 0 up to 100. Okay. And when we're born, our telomeres have about 15,000 base pairs. Okay. And then each time our cell divides, we lose about 50 to 100 base pairs off of the end of our telomeres. So over the course of your life, your telomeres are getting a little bit shorter as time goes on, as your cells divide and replace themselves. Whenever we're looking at human studies, there's a lot of variation. So those of you who are into statistics know an R squared of 0.69 is not like the best, right? But in terms of human subjects, it's a pretty significant correlation. Now we know a lot of diseases are caused by short telomeres. A lot of them are degenerative diseases. There's more and more studies coming out saying, as our telomeres shorten, these diseases become more prevalent. The classic case of the disease caused by short telomeres is a disease called progeria, and it's fairly rare, but in these cases, children are born with really short telomeres, and so they have a very accelerated aging process. Usually by the time they're in their 20s, they're pretty much old age. Right?
They only live like 5 or 6 years, or maybe 9 years.
Oh, they're definitely, especially now with our advanced medicine, there's quite a few of them that are getting up into their 20s. But still, you know, it's an important thing. So what are our telomeres doing? The telomeres are preserving the chromosomal integrity by stabilizing the genome, which is our DNA. And what's in our DNA? All of our genes, right? And what are we all about?
Genes?
Genes. Well, we're about proteins, right? And the instructions to make those proteins come from our genes.
Like the rest of the
Yeah.
world.
Okay. So they also regulate expression of genes towards the end of the chromosomes. And so as the telomere shortens, the proteins that your cells make change. Okay? And there's lots of cool studies of reversing this in mice and in cell culture. It's pretty cool. So they allow for continued cellular division. So why do our telomeres shorten? Natural processes. Okay? But they also shorten due to environmental insults and lifestyle choices. Some of the choices we make can affect what we do. Let's talk about a few of them. Stress. I think stress can age you. And if you're talking about telomere aging, absolutely. In this particular study, they measured the telomere lengths of a control group and compared that to a group of what are called caregivers. So these were people that had a disabled family member or something, and they were constantly having to care for this individual. And when they compared the two telomere lengths, then you could see that the caregivers had shorter telomeres. Okay? In this particular study, they took adults, measured again the telomere length, and compared people that had been treated poorly or were victims of violent episodes compared to children or people that had a normal childhood. And again, in this case, we're seeing a range. And this is really common, even in all of our population. If we measured all of our telomeres, we'd see all over the place. Okay? Humans are just like that. We're all highly variable. Nobody really knows for sure what it means to have telomeres that are shorter or longer in terms of predicting things. It's still stuff that's being worked out, things that are still being controlled and looked at. Okay? Another thing that people have done, again, telomere lengths, and looking at mood disorders. Okay? So again, these kinds of things somehow are affecting us on the cellular level. Okay? It's more than just what we always thought. What about obesity, diet, and smoking? You think they'd affect it? Yeah, why not? Right? Obesity does everything else. And if you're back into statistics, of course, look at these standard deviations. Huge. Okay? Again, because people are so highly variable. So you have to have really large sample sizes to do these kinds of experiments. But what it shows in terms of body mass index, the leaner you are overall, the longer your telomeres.
What's it mean by leaner? More.
Leaner, the less body fat you have.
Okay.
Okay? And as your body fat goes up, then your telomeres go shorter.
So if you lose weight, do your telomeres get longer?
That's an excellent question. That's an excellent question. Probably not.
Okay. I was like, yeah.
Unfortunately, I know. You can stop the bleeding. Okay? Think of it that way. That's good too, right?
Yeah.
Okay? Smoking, again, people who have never smoked will have the longest telomeres. Okay? Ex-smokers a little bit better. Current smokers, not so much. And again, you can see the trend is a reduction in telomere length due to smoking. Some dietary things, omega-3 fatty acids. You guys know what those are? Where do we get those? Fish.
Proteins like chicken,
Yeah, so
vegetable.
some things like flaxseed, chia seeds, there's some, okay? Mostly we, as Americans, get a lot of omega-6, right? Omega-3s, we're not so big on
I
fish
get a
eating.
lot of threes too.
Good for you, yeah. Okay, and that's a good thing, okay? Look at the difference. So here is telomere length, mean telomere length, and then in terms of how much omega-3 fatty acids, and there's an obvious increase, okay? So by losing weight, you might not, but
Eating that.
that's what, there's things we can do. Vitamin D, recent studies have just come out with vitamin D. We've been hearing a lot about vitamin D lately, right? Everybody's low in vitamin D. Sunscreen is great because it prevents sun cancer, right? Sunburn. But it's also reducing our vitamin D levels. So in this particular study, these were leukocytes, so it's really common to measure telomere length in blood because people are ready and willing to give blood for the most part. Most people don't allow scientists to take a hunk of meat and see what the telomeres are doing, right? Okay. But we're okay with giving a little bit of blood, most of us. So most of this is coming from the white blood cells. C-reactive protein is a measure of inflammation. And so in this particular graph, it's showing a little bit of both. And you can see in all cases that inflammation results in shorter telomeres also, okay? But the lowest vitamin D compared to the highest vitamin D, you can see a definite difference in telomere length. So right now, this is such a hot topic that there's labs all over the country really working on this stuff. And they're coming up with all these interesting hypotheses. Sometimes I'm like, whoa, I can't believe they found money to do that, but it's all interesting and all adds. What about attitude? It's always good to have a good attitude, right? Do you think it could really affect your telomeres?
Yes.
Yeah, it's amazing, isn't it? In this case, they measured pessimism. They had some kind of a questionnaire that you filled out, and then they took some of your blood, okay? And so here, these were the happiest, most positive people compared to the most pessimistic. Amazing. So much how we're learning that what we think and our emotions and what we're doing is affecting all of our cells on the molecular level. It's going to be so interesting. What about exercise? Exercise can cause a lot of inflammation though, right? Yeah, I guess it can. I think that the benefits outweigh the cost. What we see in studies is that with young people, we don't really see much of a difference, okay? Because there hasn't been a whole lot of telomere shortening yet, okay? But when they look at older people and compare aged controls, so people who don't exercise compared to aged athletes, we see that overall they have longer telomeres. Okay? So some things affect our telomere length positively, sometimes negatively, right? But it seems like the general gist is that negative circumstances then end up in a reduction in our telomeres. But we all know that healthy people still age, right? You probably know people that like exercise all the time, eat perfectly, right? Have the greatest attitude ever, and they're still going to get old. Okay, so there's more to it. These environmental insults and lifestyle choices then affect how quickly our telomeres shorten due to breakage. But natural processes that occur on the cell molecular level still come into play, and that's something that we can't really get around. And the natural process that gets us is something called DNA replication. Familiar with DNA replication? So what do you think is going to happen? Why are our telomeres shortening? Just because our DNA is being copied.
Some energy might lost, and you just, copy the same.
Just by mistake? Yeah, certainly. It could probably happen.
Don't we lose nucleotides at the end?
We do. So that's where the telomeres are at the end, so we lose a little bit, but why?
Because ends are left, the sticky ends or something like that.
Oh, sticky ends. Yeah, well that it's not, you know what? It makes it look like sticky ends. So sticky ends are something that we use with restriction enzymes, right? But you're right about thinking about the overlap.
Does it have anything to do, like whenever something it's heated, there's always that heat, is that excess energy? Is it anything like that?
That's a good thought. Actually, heat doesn't seem to bother DNA as a molecule too much.
No, I meant something that, that there's that energy in there. Is there any of that another chemical that has that?
I see.
molecule?
Yeah, I would say that's
A
a,
byproduct.
right. That's a possibility, which would be maybe more of an environmental insult, let's say, right? Even if it's the environment of the cell. Okay. In this case, anyone else have any?
Don't the ants just kind of wear away naturally because they're more exposed to abrasion?
That's absolutely true, which is again part of this just natural, the environmental insult kind of a thing. But there's actually then a mechanism that involves DNA replication that results in shorter telomeres, okay? And it's called the DNA end replication problem. So remember back when you learned DNA is copied by an enzyme called DNA polymerase. DNA polymerase, okay? And remember that our DNA is double-stranded, right? And it's anti-parallel, right? So one strand, one end, the other end, and the other strand's the opposite, right? So we call that 3' to 5'. And it turns out that DNA polymerase can only add nucleotides onto the 3' end of the molecule. She's like, yeah, I knew it was something. And it does look like sticky ends, you're right. Okay? So how DNA replication works is it gets started with a primer, a little RNA primer, gets started, and so on one strand, called the leading strand, of course, it just adds on to the 3' end of that RNA primer and just trucks along. The other strand, though, is a little bit different. It's called the lagging strand, and so the RNA primer starts here and hooks onto the 3' and starts, it has to keep moving its way along. And then when DNA replication is complete, the RNA primer is removed, but that end is 5'. We don't have an enzyme that can add onto it. Okay? So our blue is our parent DNA, here's our replicated DNA, and in the end, it's just a little bit smaller. Why would nature do that to us?
You kind of feel like
Because it's not perfect. I'm evolutionary. There's like an answer for everything. I don't think anybody knows the answer to this. There's speculation. Maybe this is one way to protect us from cancer cells, because cancer cells continue dividing even when they shouldn't, right? They don't ever stop dividing. But if their telomeres got too short, they couldn't divide. That's one speculation. It doesn't hold a lot of water anymore. Nobody really knows why, the answer to that. But that's a...
I have a question.
Mm-hmm.
Do cancer cells, are they able to add onto that little end
Yes.
so that they can keep going and
Yes.
going and going?
Yep, you're right. And that's where we're headed now. Okay? So what we do know is that telomeres shorten with age. Okay? And as they get shorter, then it just doesn't work out well, right? We don't want to go there. All right? Let's talk about the solution to the DNA and replication problem. Okay? There's an enzyme that's in all of us called telomerase. There's a gene for telomerase in our DNA. And what it is it's called a ribonuclear protein. It's kind of way up there, but if you see the green part, that's the protein component of the enzyme. And then it uses an RNA molecule as a template. Okay? And remember, the telomeres are the same 6-base repeat. And so it doesn't need a whole lot of engineering to start building this. Okay? And so what telomerase does is it docks onto the 3' end of the DNA, and the RNA template matches up with the telomere template, and then the enzyme, similar to DNA polymerase, adds nucleotides onto the end and extends the end of the telomere. Of course, we're still the 3' end, right? That 5' end was the problem. This is something that I had to spend a lot of time thinking about and drawing lots of pictures, actually, okay? Because it seems counterintuitive, but what it's really doing is extending the 3' end, which then the next time around when that cell divides, its telomere is a little bit longer. So the short telomere is SOL, okay? And so the telomerase adds 6 bases and then translocates and adds 6 more. Okay? Now this telomere is elongated, and the next cellular division, round of cellular division, then copies a longer molecule. Pretty cool. Okay? Telomerase is expressed in embryonic cells, which is a good thing, right? You all started out as how many cells? One. How many are you now?
Millions.
Trillions.
Trillions.
Okay? So that by the time you're born, that one cell has become trillions of cells. And what's happening each time the cell divides? Shortening the telomeres. So if there wasn't telomerase around to fill that back in during development, you'd be born pretty old, right?
Yeah.
Okay? And so it's expressed in embryonic cells. It's also expressed in germline cells, and a little bit in some of the proliferative cells. So for instance, our white blood cells produce a little bit of telomerase when they're expanding to make antibodies, you know, those. And the cells that are deep, like in our gut, right? Our gut cells replace themselves every like 3 days, right? So there's a lot of turnover. So there's a little bit of telomerase there. There's a little bit of telomerase in our hair follicles. Not enough to keep the telomeres long, but it slows down the aging process. So it's not enough enzyme to do much with. Embryonic stem cells are just like full of this stuff. It's amazing. They're such cool cells. Okay? So it's undetectable in most of our cells. Somatic cells are any cells that aren't about our sex cells. Okay? The rest of our cells are known as somatic cells, and it's basically not detectable there. Okay? And it is present in something like 90% of cancer cells have turned telomerase, turn the gene back on, and you find this protein, which is, again, how the cancer cells are managing this end replication problem. So if we think of this as the DNA, and we have the telomerase gene, and all of our genes are regulated by a sequence called the promoter. Okay? And so it's produced by the telomerase gene, and it's being expressed in most stem cells, cancer cells. It's turned on. But in our normal cells, there's a repressor, a protein that's blocking that binding site, so that way DNA replication or DNA expression, I'm saying that wrong, protein expression can occur. Okay. So what could we do? Figure out a way to remove that block, right? And if we can figure out a way to remove that block, then we could extend the telomeres of our cells and extend their lifespan. All right? Be cool. So the strategy. We know somatic cells don't produce telomerase, but we know they have the gene for it, right? It's just turned off. So what if we figure out how to turn that gene back on, extend our telomeres, extend the healthy lifespan of our cells, which then translates to us in general. Okay? So let me tell you a little bit about some of the work. Sierra Sciences, a biotech company here in Nevada, and that's their main focus, figuring out ways to lengthen telomeres. I worked there quite a long time ago now. That's me there in the top working at the hood. Can't tell, but I look much younger then, right? My telomeres were longer then. Okay. What they're doing is coming at it from the idea that if we can find a small molecule that prevents that repressor from binding and turning on telomerase, then we have something to work with. And so they're in the process of screening compounds, both synthetic compounds, they've screened a lot, and natural extracts as well. And they've found, gosh, about 1,600 now, different compounds that actually turn on telomerase. And I was there when we found the first one. It was the first one ever discovered in the entire world. And it was really exciting being there. So we have, like biotech companies have what's called a scientific advisory board. And so you pay experts from all over the world to be on your board. And then you have these meetings every once in a while and talk about what you're doing. They give you advice. And their advice was usually like, oh, I think it's been turned off so long that it can't be undone. And they were pretty much pessimistic. Okay? And science works like that. If you've done science experiments, they don't always work, do they? You have to try it again and try it again and try it again. And so this company tried it again and tried it again. They use a robotic screen. So they use robotics to do what's called a cell-based assay. And one day, compound number 57,684 was the first compound that ever was found that can turn on telomerase. So what you're looking at here is what's called a trap gel. Okay? And what this is about is you take a piece of DNA that mimics a telomere, and we put a tag on it. In this case, it's radioactive ATP. And then you take cells and you pop them open, and you collect the proteins, and you put them in a test tube. And if telomerase is there, then it will bind to the fake telomere and add 6 bases, and add 6 bases. And you let it do this assay for a while, and then you do a PCR reaction, so you amplify it, so you have lots of it so you can see it. And so what we see here, if telomerase is there and has added bases, you see, and each of these would represent effectively like 6 more bases, right? Some have only 1 copy added, some have multiples. So what we're seeing here in this trap gel is HeLa, which is a cervical carcinoma, so it's a cancer cell line, it's telomerase positive, and so we see telomerase activity, and this is just a 5%, so it's a control. MRC5 is a normal cell, it's a lung fibroblast cell, and normal cells don't express telomerase, and we didn't see that latter. So what we did is we had cells growing in culture, and we added compounds to that well in a petri dish, okay? And then looked to see if we turned on telomerase. And so you can see here in triplicate, even more meaningful, okay, that we turned on the endogenous telomerase gene. This is pretty exciting. And since then, they've done things called structure-activity relationships. They've continued to screen more things, all for the purpose of trying to, like with medicinal chemistry, hone in and make this even better, because this is pretty weak, right? I can tell you that HeLa cells are immortal, but they only express enough telomerase to maintain their telomeres. Their telomeres are really pretty short, but they express enough to maintain that. Okay? So here at TMCC, we had an opportunity to do some research, and we partnered with a group from Finger Lakes Community College, and they have an initiative called the Community College Undergraduate Research Initiative. And this has been 3 years ago now, I think, that we were selected. So you had to apply to become a member, okay? And so we were selected, and through this, we've had the opportunity to do lots of training. They've provided us with some money and some support, and it's been really exciting. So this is our team of professionals, so not any students here. This is our biology department, really. See Rachel Roeder, she's our biology administrator. Dr. Ellsworth, who's back there, was there, and Dr. Dedman. Hamid Mohammadpour works at Sierra Sciences, so he's kind of our industry contact. Dr. Bowen, who's now an administrator, he was a biology faculty member at the time, but now he's busy making sure we have what we need to keep going. That's me on the end, my hair was shorter. And Tina Sloan-Pomeroy, who is one of the lab techs in the prep lab. Okay? So we put our heads together during this meeting and came up with this idea of what our students can do. And so what we're doing here is we're doing some research that looks at how the telomerase promoter is being regulated. Okay? Because you can find small molecules and never know why they work. They just work. A lot of drugs are like that. But as we can understand what's going on with the promoter, then maybe we can create some drugs, right? Or we can do some other, maybe we can even figure out how cancer turns it on. Wouldn't that be cool? So what we're doing is we're investigating expression profiles of the protein component, which is the green part, okay, of the telomerase protein. This is a picture, if you didn't know, of chromosomes, and you can see the telomeres. And you know our chromosomes are all, there's the little centromeres. Okay, so those are little fluorescent images of the telomeres. This is a lot of stuff. I don't expect you to really pay attention to it. But what we've got is DNA sequence of a portion of the telomerase promoter. And other research has identified DNA-based sequences or spots in it where transcription factors would bind and affect regulation. So a repressor would be a transcription factor. It turns it off. Okay? Activators or enhancers would be transcription factors that turn it on. Okay? And so you can see here in this part that we have EBOX sites, SP1 sites, AP2. Those are sites where transcription factor proteins bind and turn on gene expression. And our students, this first time around, decided to look at a MYC binding site. And, you know, we kind of like talked about all the different kinds, and this is what they wanted to do. MYC's kind of pretty hard, okay? Because MYC doesn't do anything by itself. There's a MYC-MAD complex and a MAD-MAX complex, and sometimes they're a repressor and sometimes they're an activator. So I think teasing out this is going to be tricky, but that's what we did. What we're doing is we're using what's called a promoter-reporter assay system. Okay? And in this system, we use a circle of DNA called a plasmid. Are you familiar with plasmids? Plasmids are actually bacterial DNA.
Bacterial DNA.
Yeah. Bacteria actually share them with each other. And antibiotic-resistant genes are most often on these plasmids. And that's how bacteria share antibiotic resistance. That's why you should take all of your antibiotics to make sure you kill those bacteria. Because if one of them survives with the plasmid, it shares it with everybody. Okay? We're taking advantage of this as molecular biologists. So what we've done is telomerase is a hard enzyme to assay. So we put in a reporter, which is much simpler. It's called secreted alkaline phosphatase. So that's our gene. It's pretending to be telomerase for the purpose of what we're doing here. And then the promoter, what we do then is we mutate the original sequence in the promoter. So you can see here we've changed this G nucleotide to an A and a T to an A. Up there we've switched a CAC to a TTT, which should effectively prevent binding of the transcription factors. And if the transcription factors can't bind to the promoter, then you don't see normal gene expression. Okay? And we take these plasmids then, and we insert them into mammalian cells. So we're growing cells in Petri dishes, and then we actually pop holes in the membrane and kind of like encourage the plasmid to get inside. And the plasmid gets inside and actually then is transcribed and translated into our reporter protein. Okay? So we measure promoter activity by following seep levels. If we have a lot of seep signal, then we have more promoter activity. And so by doing these assays, we can look and see, does this mutation cause a problem? Is this an important part of the promoter for telomerase? By seeing whether or not we lose or gain activity. Okay? And we're going to use that information then to try to learn more about what's important. So this is some data from our students. In this case, we used cancer cells, and it's units of luminescence. Secreted alkaline phosphatase is an enzyme, so it's a protein. And what it does is we've put in a substrate, another molecule, that this enzyme can effectively bind and break apart. And so if the enzyme's there, it breaks apart this molecule, and when it does, that molecule releases a photon. And then we use what's called a luminometer to measure the photons. So the more photons we see, the more enzyme was there. Does that make sense? We make sure there's plenty of molecules so it's not the rate limiting factor. So when we looked at cancer cells, what we've got here, empty vector, you always want a negative control, right? So this is plasmid but doesn't have any gene. It doesn't have gene in it. The SV40 promoter is our positive control. Not looking so good here, is it? Okay. Yeah, that particular plasmid wasn't so good, but that's the way science works. It's really a good plasmid. We had some problem with our assay. Okay. HTERT is the telomerase minimal promoter. And is telomerase expressed in cancer cells? Yes. So would you expect to see SEEP activity in a cancer cell using the HTERT promoter? Yes. Site C deleted is something CR Sciences came up with. They discovered a repressor site, and if you mutate that part, then it relieves repression. Okay? So it's not that important in cancer, but in normal cells it's more important. Okay? So these two are effectively about the same. Here's where we deleted those two mix sites. And in cancer cells, we had a pretty big increase in SEEP activity. Okay? We don't know what that means yet. We've only done this experiment really once or twice. So it's an ongoing project. This is like hot off the presses data. We also did it in a normal cell called a BJ, which is a foreskin fibroblast. Look at the scale here going up to 50,000. Look at the scale here going up to 80. I would never really do this. Is this a good way to present your data? It kind of gives you a false impression, doesn't it? It looks like you got a whole lot of activity. But really, our activity is like right about this flat line, right about here. Effectively, it didn't work. Okay? So we were like, oh, great. Right? Bummer. So what we had to do is a bunch of experiments, and what we learned is this type of fibroblast is not taking in the plasmid. So plan B, we tried another normal cell. Okay? We tried a lung fibroblast, and in the lung fibroblast, you can see we're at 7,000 at least. Okay? 100-fold more than the last one. And here we're seeing more what we're expecting. We have our empty vector. We have our positive control. Here's our telomerase promoter in a normal cell. It's repressed. delete site C and we've got it. And look what happened when we deleted the E boxes in normal cells. Nothing happened, yeah. So we have a lot of work to do. Our students are working on figuring out what to do next and kind of tease out more information. But this is the two students that have been working on it, Sammy and Melissa, and they just recently went to Phoenix to a Curry conference, which was our partner, right? And presented their data as a poster, and I didn't end up going, but I understand that it was quite popular and they had lots of activity at their poster. Okay. So that basically concludes what I wanted to share with you. There's so much cool stuff, but you kind of know a little bit, right? And in case you didn't know, our governor declared September 22nd through the 8th is Telomere Research Week. So telomere research has hit the big time. Is there any questions?
What can you tell us about the mice?
Oh, about the study? So this been working on this forever, okay? So what researchers did, mice are different than humans. They actually have really long telomeres naturally, and they express telomerase. Yes, let me finish. So what they had to do there, because we don't have a good model to study this, okay, so they engineered mice then to have telomeres that would age. Okay? They did this actually by taking the mouse telomere out and putting in the human. That's a good model, right? And so now these mice would age like you'd expect. And so then what they did is they put in, it's kind of like gene therapy treatment, which is we're getting better at, but we're still not really good at gene therapy. But we're getting better. And mice we can practice on. And so they used gene therapy to reintroduce telomerase into those mice. So they had litter mates with all short telomeres. Part of them they did nothing to. The other part they added in the telomerase gene and saw this actual reversal of aging. It's not the first time it's been done. It's been done in the 1990s when all this was first discovered, but not with human type things. It was done much simpler way, you know. So this is the first time that we've actually shown in a real live organism that this really does work. Okay, so that was a really well engineered, amazing experiment that took years and years, but gave us the best evidence that we have that it's a good thing. Okay?
So why do mice have longer telomeres than humans?
Yes, they do naturally, and why? I mean, I think it's part of their DNA makeup, right? Evolutionarily, they are different than us. Hey, mice age differently. So mice have long telomeres, but they're not very efficient at dealing with antioxidants. It's kind of a complicated way. So they don't deal with free radicals, oxygen radicals anywhere near as well as we do. They're lacking some of the enzymes that we are. Superoxide dismutase is one of them. And so they need to have these really long telomeres because they're getting bombarded all the time. Okay?
In other words, they're born that way.
They're born that way, absolutely. And that's part of their species. It's not a good answer to your question.
So that's why they're not hooking humans up with mice telomeres, right?
Right. We've actually done some studies where we tried some things like that. But yeah, that's like, none of us want mouse telomeres. We want human telomeres that are just naturally long.
So if we were to able to block or unblock this receptor site, and we were to able actually to produce telomeres, do your cells divide faster also as you're younger? Because it seems like you would also, if you would elongate your telomeres, then you would also then, I feel like you would start to reverse your aging, like you're saying.
Oh, so yeah.
It just doesn't seem to make sense because it seems like you could almost go backwards in time.
Right. And so there's absolutely been studies They did a study where they took human skin and put it on the back of a mouse and then treated that skin with telomerase, and that skin became young again. Not yes, yes. And what it has to do with, probably, because they also did gene expression profiles between young and old, and the gene expression, so the proteins that are in young cells are different than the ones in old. And so as the telomere shortens, it affects what genes are expressed or not expressed, right? So it's way bigger picture than probably any of us understand yet. And it's more than just this long telomere, you know, protecting the integrity of the genome. Okay, you had a question?
Oh yeah, sure. In a while. So it's a common question, like, you know, when who does not understand all the biological terms.
Mm-hmm.
So the common questions is like, how long is it going to take for the biologist to, you know, reverse the process so that we cannot be old again?
Right.
In other words, can we do it before we get old, so that, you know, we don't have to face all that?
Yeah. So,
So like, where are we at this point? Are we like far, or we just started?
you know what? Science is just beautiful that way, right? Okay. Who knows? Who knows? Okay. Science is like on this precipice, right? It's like one more experiment and you've got it. Or not, or not, or not. Okay, so that's like a easy answer for me to just kind of blow that off in a way. But the reality then also is the more money, the more effort, the more people doing this, the quicker things like this can happen, right?
Maybe more development we have in this field, or more technology we're using.
Absolutely, and not even in this field, but in the field of delivering proteins to cells, right? In the field of detection. So all nanotechnology, all of the different aspects all feed into all of these pictures, right? Okay? So I don't know. Yes, is it possible? Absolutely, yes.
Yep.
Absolutely possible. In your lifetime, possible in my lifetime.
Oh yeah, in the meanwhile.
Yeah, absolutely. So I'll go back to you.
What about?
We'll go back here, sorry.
How old do you estimate us humans as a species will be able to live if we can remove the blocker?
Good question.
Especially when you're 10 years old.
Especially when you're 10 years old, yeah. So yeah, the sooner the better. That always is true, okay? And it depends on expression levels. Okay, first of all, it won't prevent you from ever dying, because if you get hit by a bus, it doesn't matter how long your telomeres are, okay? And there's other things that cause us grief too, right? So, but I can tell you this. Cancer cells have been dividing in laboratories for 70 years, okay? And are not slowing down. They're fine. We consider them to be immortal. Okay? So in a big picture, if everything was perfect and that's all there was to the story, then probably a long time. Right now, what they figure is, given what we have now, the best we can probably do is 150 years, but nobody even makes it that far because of other problems. Okay? So theoretically, I guess you could keep on going. So long as something else didn't get you.
Oh yeah.
Right?
A bear.
Like a bear, absolutely. A bear, brutal. Okay, I'm gonna go here.
What about telomeres and animals that live longer than humans?
Oh, that's a really good question, because there are animals that don't appear to age, actually. Like tortoises and lobsters. They don't appear to age, they get bigger.
Small too, just.
They get bigger, but if you look at their gene expression, it's not really much different. They're not really aging. So not all species age by telomere shortening. We don't know why. Okay? And so there's different mechanisms going on in other species. I mean, we can even think of bacteria, right? Bacteria are basically immortal. Okay, so who's I skipping people?
I was just curious, you said that telomeres are responsible for gene expression. Are they the sole responsible?
Absolutely not. Absolutely not. So what that's meaning is, and probably it has to do with how the DNA molecule folds back. Okay? So the telomere is this long molecule of the same thing, right? The same repeat. And it can fold back over on itself and proteins bind it. And so it forms like this cap. Okay? Right, yeah. So it's got a lot of stuff in there. All right? And as it gets shorter, it's not able to fold over this cap. Okay? And so the idea being that this folding over probably affects gene expression on genes further upstream. And in some cases, genes are affected by regulatory sites, thousands of base pairs upstream of the gene itself. And so it hasn't actually been teased out what's going on. There's just hypotheses. But it probably has something to do with this way that the telomere forms this cap on the end. Okay?
It seems like if we were able to do this, you would want to stay almost at a certain age, because if you were producing the proteins that you were making when you were a 4 or 5-year-old, you're not getting the same brain stimulus, and you would want to like find the peak of what our age would be.
So what you're asking me, in a way, is, what was that movie with Brad Pitt? Yeah, right. You know, so even though that's really extreme, but you're not gonna, you would not lose what you already have.
But are there proteins that when you're young you just, like, I'm thinking brain cells, honestly. So like, when you're making those proteins at a young age, they're not, I'm just trying to.
Yeah, no, it's like amazing to think about, isn't it? Yes. And so you would still want your genome to be expressing what it's supposed to. And there's no reason to believe, because your telomeres wouldn't keep growing forever, because you're still losing them, right? So there's no reason to believe that you would actually lose what you've got or go backwards. Like all of a sudden start producing a whole bunch of human growth hormone and you start like growing like crazy. There's no reason to believe that that would happen. And they're not seeing it in animal studies and in cell culture gene profile studies. Exactly. Does anybody want to be the first guinea pigs to like, not me. No, I want a little bit more. Okay, there was someone, okay.
Are we sure that the telomere shortening is what actually causes the aging, or is that just another result of some other factor that actually causes the aging?
That's a good question. And what I can say is science doesn't really prove things, right? We're always learning something new that kind of like changes or maybe even cancels out what we thought was true before. So from that regard, I don't have a perfect answer.
So I'm not really sure that telomeres are what cause the aging.
What I can tell you is that when telomeres shorten, then cells age. And when you lengthen them back up, they get young again. So whether there's more mechanisms, like for instance, and it probably is quite possible, that long telomeres cause the expression of something that keeps you young, right? Kind of like what you were saying.
Factors too, you're talking about stress and all those things.
Right, there's a lot of things that come into play, absolutely. So there's a lot of things that cause aging, but it's pretty clear that telomere length has something to do with it. And keeping them longer is better than letting them get short. There's still a lot of things to learn though. Okay, you go, someone has to ask a question.
How has the development of receptor unblockers and the success in finding compounds that are unblocking it, how has that opened up the field? Are there other agencies that have jumped on board?
Absolutely. So now after we've started like publishing that this could actually happen, of course there's lots of people. And if you Google like drugs to keep your telomeres long, you'll get choices, all right? They're out there, okay? But the whole idea of the more we understand about how we can deliver drugs to cells and how we can affect how cells respond, all comes into the big picture, okay? So it's a little bit different in terms of receptor-mediated type processes, But any information we get from that absolutely translates to one new idea that makes something better. Okay?
You mentioned that there was a nanotechnological component to this. Could you maybe go into a little bit more detail of what you mean?
Sure. So in a general sense, nanotechnology is it's working towards being able to deliver things much more effectively. Okay? Right now, if you take a pill, what happens? It gets into your stomach, right? It ends up in your liver, may get changed. Some of it ends up in your bloodstream, okay? So the better that we are at delivering things like through nanotechnology, and so through nanotechnology, they're working on making little bots, right, that the drug could be in. And so it's about delivery, okay? There's other ideas that have come on, like, so instead of trying to turn on the endogenous telomerase and extend the telomeres, what if we could just deliver telomere sequence and tack it onto the end? Okay? There's people working on that. Okay?
You said our somatic cells don't have but they have the gene.
Correct. Correct. Did you have a question?
Yes. I was wondering if you're aware of any health problems that those younger mice might have experienced during the project.
No, but it's a couple years old. Mice don't have a very long lifespan, but no, I'm not aware of any problems that they've had. And when we telomerize cells in culture, so when we insert the telomerase gene in, those cells, for the most part, keep the same phenotype. They don't turn into cancer. I have seen some turn into cancer, but I've seen regular cells turn into cancer too. So all of our cells, right, can become that. I haven't seen that adding telomerase in predisposes them to getting cancer. Okay?
Okay. So we know that some things we cannot control, but some things we can, right? Like health issues and attitudes and stuff.
Right.
So is it possible if anyone controls them perfectly or to some extent good, we can, we cannot go backwards, but we can kind of, delay or aging?
Yes. Yes.
And my second question is there anything related? Correct me if I'm wrong. The gene mutations or genetic behavior has to do something with telomeres, and we can use that genetic behavior to, elongate telomeres rather than shortening them.
Right. So yes, there are some cancer cells that don't use telomerase to extend their telomeres. They use something called recombination. Okay? And so in recombination events, instead of actually an enzyme extending it, then chunks from other chromosomes or get added on. Okay? And so it's not good for us. It won't work for us. But some cancer cells use that. They're so messed up anyways.
Oh, yeah.
Right?
So we were just talking about the nanotechnology and putting it in a local place. Could this also be for after surgery or something like that?
A transient kind of, absolutely. And probably in the big picture, that is going to be probably what works. So you don't turn it on permanently, okay? You do it more like, you take it for a while, extend your telomeres, you stop, you know what I mean? Ideally, that's probably what would happen. Yeah. Okay.
Were you saying that there was research being done about creating repressors to and if you could do that and direct it into cancer cells?
Yes.
I was wondering about if there's any research.
Yes. So I see what you're saying. So obviously there's a repressor protein. What's happened in cancer cells for the most part is instead of, they might still be regulated normally at the endogenous gene, so the original promoter gene series. What happens in cancer cells is the telomerase gene often gets copied and inserted in front of other promoters. So in other words, the HeLa cell, the cancer cell I showed you, has like 13 copies of the telomerase gene dotted throughout the genome. So what cancer's doing, and they haven't studied all cancer cells, but the ones they've studied so far, they tend to have, so it's kind of like a hot spot where it can jump and end up in different spots of our chromosome, and then be under the control of a different promoter that is not regulated. So so from that idea, the repressor wouldn't necessarily work if it didn't work on all promoters. Does that make sense? But for anti-cancer, there's some really cool things going on. There's a vaccine that's in clinical trials and is being used for prostate cancer with some really good results. And it's a vaccine against cells that express telomerase. And so it kills cancer cells kind of preferentially. Okay? That could have some side effects, of course, for our blood cells and things like that, right? And so then you work towards being even more specific. Okay? Some other ideas are what are called poison vectors, right? If telomerase is expressed, then instead of SEEP, what if you put a toxic compound in that plasmid and then introduce that to cancer cells? And if telomerase is active, then that poison gene gets expressed and kills the cancer cells, but normal cells don't express it, so they're okay too. So there's a lot of stuff going on, and as many ideas as you can think of, I mean, it's like all over the place.
Sky's the limit.
Yeah, I mean, why not, right? That's what makes science fun. No such thing as a crazy idea, as long as you got money.
What would be your recommendation for somebody who was interested in bridging the gap between where they are now in their studies and getting into the field?
Oh, good question. So, as you can see, you saw my path, right? I didn't spend any time researching telomeres and telomerase until I actually got a job, okay? So in terms of that, the more you can learn just about techniques in general to make yourself really marketable when you go, you already have an understanding. It's a lot about the understanding too. So I can teach even a non-scientist to do an assay at a bench, right? They don't have to know what's going on or why they're doing it. But keep digging into asking the questions why. What's happening behind the scenes, okay? And so from that regard, math is super important, okay? At Sierra Sciences, when people would come in for an interview, we used to give them a math test. And you were judged, okay? Because math is critical. We're always like mixing reagents or even little things like someone was just, what did they want to do? They were doing what's called a transfection. And so they were trying to introduce multiple types of plasmid DNA into the cell, and they wanted to have not equal molar, but equal copy number. Okay? So you had to figure out the molecular weight of all of the plasmids, and then you had to figure out, based on concentration, so we're always working with math. Of course, the robotics aspect of things and the biomedical engineering stuff is always about math. So math is critical. Okay? A good understanding of basic science, biochemistry, and molecular biology, as well as just a general biology is a good solid foundation. And then depending on where you want to go, right? If you're interested in the nanotechnology fields, if you're interested in the robotics or detection of detecting things that we can't even see now, okay? I mean, then you're more into the engineering. At Sierra Sciences, when I worked there, we hired biologists from UNR, biochemists from UNR. We also hired chemical engineers. And so we had a few chemical engineers, chemists. We didn't hire any strict mathematicians, but if one had applied, I bet we would have. Because the president there is so much about math.
Mathematicians like to prove things rather than getting more assumptions.
And yes, and scientists are a little bit more emotional maybe than strict mathematicians, engineer types sometimes, right? And so it's always good to have this good mix. And the more ways you come at something, the better, right? Okay, I don't know if that did a good job of answering your question, but math is important, and then the other stuff kind of comes together. If you have a good foundation, math, chemistry, and principles of biology, you're good. Okay, I know we went a long time. Is there any other questions? Well, thank you for your attention. Congratulations on a great club.